Welcome back. We are starting with chapter 3, and we will talk about the bacterial cell structure. We will cover the section 3.2 through 3.9 in this chapter, just not covering the 3.0, the controversy about the prokaryotes that we just briefly touched. So in the 3.2, the bacteria are diverse but share some common feature. And what we are going to do, that we are just going to look at the shape, arrangement, and the size. So the shape, the cocky rod are the most common, and the cockies are spherical. Rods are the bacillus, which is elongated, and various other. We are going to talk about the arrangement, and the arrangement is determined by the plane of division, that whether they just divide in one plane, that will form chains. If they were divided in many different planes, they will form clusters, and so on. And the size also varies. The cocky shape and arrangement, so the coccus is spherical, and the cocci is plural, and they are just as similar, that we are seeing that these are some of the coccus, these are some of the coccus or cocci. On the other hand, these are actually rods, so you can just distinguish that how it is different. So the diplococci is in pairs. Streptococci, if they are growing in chains like this, these are streptococci. We are going to talk about that. Streptococcus singular, streptococci plural. If we just have a bunch of grape-like structures, those are called the staphylococcus, which are divides in many different planes. If we just have a specific 4 cocci in a square, 1, 2, 3, 4, they are tetrads, and Sarcini in a cubic configuration of 8. So they can divide in 2 planes, and they are forming the cubic formation, which is called the Sarcina singular or Sarcini in plural. If we just look at the other shapes and aggregation, the bacillus is singular, are rod-shaped bacteria, very, very simple. And the coccobacilli are actually very short rods. On the other hand, the vibrios actually resemble rods, but sometimes they are kind of a little bit crooked, which is comma-shaped. On the other hand, the spirula, singular spirulum, is rigid helices. What we mean by the rigid helices, that these are some of the stratic structure, and they could be like a spiral shaped, and if they're alive or dead, they're going to be in that shape as well. That we mean by that is a static structure. On the other hand, another group is a spirochete, which is a flexible, not rigid, flexible helices. And these bacteria are in constant motion. As they move in a corkscrew-like motion, they actually produce this type of structure. So this is a dynamic structure and of a flexible helix. Now, if the bacteria is dead, they are not going to form this type of structure. It's going to be a long, simple rod. So this is a dynamic structure, and the movement caused this spirochete to have the flexible helices structure. On the other hand, these are the rigid rigid helices. On the other hand, the mycelium is actually a network of long multinucleate filaments. So each filament, say for example, each filament is known as a hypha or hyphae, plural. When there is a mass of these, then you can actually call them as a mycelium. So mycelium is a network of long multinucleate filament. Each filament is called a hypha. So, and these hyphae all together form a mycelium. This word is actually very common in the fungal. When we actually just talk about the fungus, we will talk about that. But these we use for some of the bacteria like actinomyces. Some of the cyanobacteria also form these kind of mycelium photosynthetic bacteria. And you can see that these are some fruiting bodies. There are different types of bacteria that you can easily see. Streptomyces also form the filamentous bacteria, just like forms the mycelium, and this is another type. Pleomorphic organisms that are variable in shape, and some of the bacteria actually do have many different types of shapes. We call them as pleomorphic. We are going to talk about that either. So here is a size comparison. You can see that compared to a red blood cell, an E. coli is much smaller, about 1.3, 2 to 6 micron in diameter. Streptococcus is much smaller, about 0.8 to 1 micron in diameter. And you can see that they are considerably larger than all the viruses, and that's the reason we can visualize them under the light microscope, whereas the virus, we cannot usually see them except just probably 1 or 2 viruses without the electron microscope. Now, the smallest of all these is a mycoplasma, about 0.3 micrometer, and they do not have a rigid cell wall. And that's the reason that they are called pleomorphic. That means they can change in the shape, and this is one of the smallest bacteria that you can have. On the other hand, this very large bacteria, this Epulopiscium fischeloni, you can easily see that they are 600 micron in length and about 80 micron in diameter. One of the giant bacteria so they come in all different shapes and sizes that you can appreciate. Then, if we just look at a size and shape relationship, what does all that mean? This is actually a very important factor in terms of surface to volume ratio, because this actually depends that how the bacteria is so successful in the survival. So if the organism is very tiny, you can see that this is R equal to 1 micron. The surface to volume ratio is about 3. As it is becoming bigger, the surface to volume ratio is becoming 1.5, so it's decreasing. So smaller the size, the chances of that organism to pick up the nutrients from the environment is much higher, and that's why they are so successful. On the other hand, there are some advantages, although not nutrient uptake. Large size or odd shape may be protective mechanism from predation. So this is clearly an advantage of this E. fischeloni, which actually is very, very big. So the problem is that they have to compromise on the nutrient uptake, but they actually do have the previous bacteria that we saw, that E. fischeloni, actually circumvent this because they actually have invagination of the plasma membrane under the cell wall to increase the surface area of absorbing nutrients from the environment. So they can so these bacteria actually are also successful because they can actually circumvent the problem of the large size. So next we are going to talk about the bacterial cell organization and the cell envelope in 3 layers. Then we're going to talk about the material that is present in the cytoplasm and the external structure. So this is a common bacterial structure in their function. You can just, this is very self-explanatory. This is just for your review. You can look at that at your own time. So in the first part of this is the bacterial plasma membrane, which control what enters and leaves the cell. Now, we are not going to talk about the plasma membrane in detail because we have covered that in Biology 8420. So we'll just briefly mention just a couple of things about the plasma membrane. So just by definition, it encompasses the cytoplasm, absolute requirement of living organism. And it is a selectively permeable barrier. It can decide what it will allow to pass through and what it will not. It interacts with the external environment because it does have the receptor for detection of response to chemicals in the surrounding, and they can move towards this chemical, which is called the chemotaxis. They also do have transport systems like channels, different types of protein channels, and different transporters, which actually can get material from the outside world or also can expel material from inside the cell. And the bacterial plasma membrane is also responsible for various metabolic processes because as they do not have organelles, many of the enzymes like respiratory enzymes, DNA replication enzymes, they are associated with the plasma membrane and help in the metabolic processes. So the next, what we are going to talk is the bacterial cell envelope. The plasma membrane is not required in detail, and we're going to talk about the cell wall and layers outside the cell wall. These all together make the bacterial cell envelope. Let's dissect this one at a time. Before going into the detail of that, just one thing that I want to just mention out of the plasma membrane, which we are not covering, that microorganisms require iron. And the ferric iron is extremely insoluble and difficult to actually obtain. So what they actually do, that the microorganisms secrete an organic molecule, these are known as a siderophores. This siderophores actually complex with the iron. So this is the iron, and you can see right here, this organic molecule actually is coordinating that iron, and this complex with the ferric iron, this siderophore, can be transported by the cell by specific transporter, and this is the way that they can actually uptake the iron through the plasma membrane. So this is one of the important things that please remember this. So next we are looking at the 2 main types of bacterial cell wall that we have talked about in the staining process, the gram-positive and the gram-negative. So before that, let's take a look at that the function for the cell wall, it maintains the bacterial shape, protects from osmotic lysis and toxic materials, and sometimes there are components on them which can contribute to pathogenicity as well. So the peptidoglycan, which is also known as murine, is the unique material that is present only on bacterial cell wall. It is not present in archaea and it is not present in any eukaryotes. This peptidoglycan, you can see that it's made up of peptide, and glycan means carbohydrates, is a rigid structure outside the cell plasma membrane, forms the bacterial cell wall. And there are some variations of this. We are going to talk about that. On the other hand, the two types of bacteria based on gram stain that we talked about, the gram-positive, which stain purple, actually have a much thicker peptidoglycan, whereas the gram-negative bacteria, which stain pink or red, actually do have a thin peptidoglycan and the outer membrane. So there are some other differences as well, and we're going to talk about those in a moment as well. So let's first take a look at the peptidoglycan structure. So the peptidoglycan structure is made up of these two alternating modified sugars, which are derived from the glucose. The first one is N-acetylmuramic acid. There's a carboxyl group in here. That's why we call it an acid. Whereas this is an N-acetylglucosamine, where you have an amine group right here. And that's why we call this N-acetylglucosamine. These two are bonded right here. And these are the alternating molecules. So what you are going to see, you're going to see a NAM, NAG, NAM, NAG, NAM, NAG. And this chain is elongated and round, and it will just go on like this. So these are the backbone of the peptidoglycan structure. So from this backbone of the peptidoglycan structure, there are some peptides attached to it. So these peptides are called stem peptides, which are made up of several different types of amino acid. So these amino acids actually can vary in different bacteria. So a typical one, if we just see in this example, you can see that there is an L-alanine, L isomer of alanine, added to a D-glutamic acid, is a D isomer of glutamic acid, both of them amino acids. Then this is a mesodiaminopimelic acid, or DAP. Then you have another D-alanine, another D-alanine. Now, the interesting point that I just want to mention in here, that in eukaryotes, in other organisms as well as us, all the amino acid that is present in the protein and that we can digest and utilize, they are all in L form. They are all L isomers. So in human beings, in the proteins, we have L-alanine, L-glutamic acid. These are all L forms. In the bacterial stem peptide, we only see this D-glutamic acid, or the D isoform, and this actually does have an advantage. And this is one of the amino acids which is not present in the protein as such. So the advantage of these D amino acids are that if the bacteria encounters is a pathogen and it goes into an organism as a pathogen, and if the host to whom it is actually infecting produce proteases, peptidases, which will digest these, then it is a problem for the bacteria to lose the battle. So what happens that the eukaryotic proteases or peptidases cannot digest or recognize these D amino acids. That's one of the protective nature of the peptidoglycan, which actually help them to survive in a protease or peptidase if secreted by the hosts. So here there are two examples that how these NAM and NAG, and they are actually cross-linked and do have a helical shape. So when they are cross-linked, there are two types. Say, for example, the top one is very common in E. coli, where you are seeing that there's an L-alanine, D-glutamic acid, diaminophenic acid, and D-alanine. And in the previous picture, you have seen that there is another D-alanine. When this D-alanine forms a direct linkage with the diaminopimelic acid of another backbone strand, so there are layers all over. This is one layer, this is another layer, and you have a cross-link. Then the extra D-alanine which was here and the extra D-alanine which was here is actually take part in the reaction and go out. So if you remember that right here, you have two D-alanines, At this site, a bond may be formed to link this peptide to one of the another chain. When this occurs, the terminal D-alanine is actually lost. That's why you do not see that terminal D-alanine right here and the terminal D-alanine here. This structure is more common in a gram-positive bacteria like a Staphylococcus or something like that. And what you are seeing that the composition of the stem peptide is a little bit different. As I said before, it may vary a little bit. Here, the first one is an L-alanine. Then you have a D-aminated glutamic acid. So here you have an amine group near the alpha carbon of the carboxyl end, which is an extra one. So this is a modified amino acid. Then you have an L form of lysine, and you have a D-alanine as well here. And you have the same thing right here with the other chain. Now, what you see here, this D-alanine is actually bonded to the L-lysine. See that DAP, the aminopamelic acid, is not present in here. But this D-alanine and L-lysine is actually connected together with this peptide interbridge. And these are 1, 2, 3, 4, 5 glycine. So this is called a pentaglycine interbridge. So these are the typical type of cross-linking that we actually do see in different types of bacteria. And these are some of the targets of some of the antibiotics like penicillin that we are going to talk about them in later part. So what happens, all these cross-linking gives the cell wall a solid, rigidity. Along with that, they may also contain these teichoic acids, which are negatively charged. And these help to maintain the cell envelope, protect from the environmental substance, and also bind to the host cells. And some gram-positive bacteria have layers of proteins on the surface of the peptidoglycan as well. So if we are looking at the gram-positive cell wall, right here, this is the cell, this is the plasma membrane, and you have a periplasmic space after that, and then you have the thick peptidoglycan layer. Now, the teichoic acids are present only in the gram-positive bacteria, only in gram-positive, not in gram-negative bacteria. And what they are, the teichoic acids are actually made up of glycerol or ribitol, and these are actually attached with phosphates, glycerol or ribitol phosphates. And these teichoic acids actually can be two types. If the teichoic acids are attached to the peptidoglycan, we call them as wall teichoic acid, wall teichoic acid. If they're attached to the plasma membrane, we call them as the lipoteichoic acid. And we're going to see them soon in somewhere else. So here, the periplasmic space is actually mentioned. It's illustrated right here. So this is the plasma membrane right here. This is the cytoplasm, plasma membrane with the peripheral and integral proteins. You have the periplasmic space. On top of that, you have the peptidoglycan layer. This is a typical gram-positive bacterial cell wall. Here you can see these are the NAG and the NAM molecules, multi-layered, and they are all cross-linked. And you have two types of teichoic acid. You have wall teichoic acid, as I said before, cross-linked with the peptidoglycan, wall teichoic acid, whereas the lipoteichoic acids goes all the way and inserted into the phospholipids of the plasma membrane. The periplasmic space actually do have some exoenzymes secreted by the gram-positive bacteria, and they actually help to break down large nutrients. The periplasmic space actually is filled with many of the different types of exoenzymes which are secreted from the cell, and they help to degrade some of the larger nutrients before they can pass through the plasma membrane. So the gram-negative cell wall, on the other hand, is actually much more complex. So if we just look at the typical schematic, that the plasma, here is the plasma membrane, then you have the periplasmic space, and then we have a thin peptidoglycan layer. This is very different from the previous one. If we just look in this structure right here, you can see that it is a huge, big, thick peptidoglycan layer, whereas in the gram-negative bacteria, the peptidoglycan is much thinner. On top of the peptidoglycan layer, there is an outer membrane composed of lipids, lipoprotein, and lipopolysaccharides. So this is called the lipopolysaccharide or the LPS layer or the outer membrane. Very different and only present in gram-negative bacteria. There is no teichoic acid present in gram-negative bacteria as well. So if we just look at the gram-negative cell wall, the outer membrane actually is the outside of this thick peptidoglycan layer. So you can see right here, this is a thin peptidoglycan layer, and there are some adhesion proteins, and you can see that these are called as a bronze lipoprotein. This bronze lipoprotein actually help the peptidoglycan layer attach to this outer polysaccharide layer, outer membrane layer, which is actually made up of different types of lipoproteins as well as lipopolysaccharides. The peptidoglycan is approximately about 5 to 10% of the cell weight, whereas compared to a gram-negative positive cell wall, the peptidoglycan will almost have 90% of the cell wall weight. The periplasmic space also differs. It may constitute about 20 to 40% of the cell volume in gram-negative bacteria. Many enzymes are present in a periplasm, hydrolytic enzyme, transport proteins, other proteins, and there are many enzymes and transporters which are present in the outer membrane, which actually help even or act as antibiotic pumps as well. So we're going to talk about one of these. This is a large protein called PON-B, which actually have an outer membrane receptor and attached to the inner plasma membrane component. And we are going to see that this actually helps or aids in transfer of molecules by some specific mechanism. So other than that, what you are seeing, that there are these trimers, these are the porine molecules, and these porine molecules actually are hydrophilic channels which actually transport a lot of different types of material that we are going to see in the next slide. So if we just look at this lipopolysaccharide structure, typical, so it consists of three parts. So the first part is the lipid A, and this is actually buried into the outer membrane lipids as such. So this is called the lipid A. So the next part that what you have is the core polysaccharide, and the core polysaccharide has several different carbohydrates attached to that. So these are glucosamines. Then you have the 2-keto-3-deoxyoctonate, and then you have the heptulose, and then you have, this is the O side chain of the O antigen. So these are the three different parts, and the core polysaccharide and the O side chain extend out from the cell. So this is the outer part, and this is the anchor. So the next part that what we are looking at, that the LPS, or the lipopolysaccharide, contributes to the negative charge on the cell surface, helps to stabilize outer membrane structures, permeability barrier, and right here the O antigen is a defense protection, or this is actually called an O antigen as well. So this lipid A parts is an endotoxin. So what happens that when the gram-negative bacteria actually die, their cell wall actually disintegrate, and the lipid A actually can so when the bacteria die, gram-negative bacteria die, the lipid A can actually disintegrate, and it can actually react with the neutrophils and have hyperreactions, and which we call as an endotoxic shock. That is possible after taking antibiotic in a severe bacterial infection. We're going to talk about that somewhere else. So the gram-negative outer membrane permeability is more permeable than the plasma membrane due to the presence of these porine proteins and transporter proteins. So the porine proteins actually form a channel. So this is a channel, this is a channel, this is a channel, this is a side view. You can see a beta barrel-shaped structure made up of these beta strands. And so these channels are actually water-filled, and the molecules up to 600-700 Daltons molecular weight can pass through these channels. So they actually are always in a trimer shape, and this blue protein actually helps to keep all of them, all the three trimers together. And what you are seeing right here, this is the small orange loop that you are seeing in the middle. They actually can tighten or make this channel smaller or larger. So those are actually essential for allowing certain molecules to pass and certain molecules to block. So these are the regulator proteins to maintain or to regulate the channel diameters. So this is a typical porine, which is trimer with 3 channels. Now, other than that, in a couple of slides back, we have seen that this is a ton B, and these are some of the other type of proteins, and these are also transport proteins. And let me show you that how this actually works. There are some very larger nutrients which actually cannot enter the cell with these porine proteins. They're too small. These are larger than 600, 700 daltons. So what happens that when they come in contact with the outer membrane receptor of the ton B complex, this actually triggers right here an energy transduction. So you can see that is attached. So there is an energy transduction process goes on, and that energy transduction process helps this large molecule from here to enter into the periplasmic space. So once it enters the periplasmic space, then those molecules, after coming into the periplasmic space, can get into the cell by these ABC transporters. These are known as the ATP binding cassette transporter. We have talked about that in cell biology. And so this is a 2-step process that very, very large nutrients, which are not possible for the porine to get in, they can actually get through these large transporter by 2 mechanisms, which is known as ATP binding cassette transporter proteins, along with the ton B complex, which works by the energy transduction mechanism from outer lipopolysaccharide layer to the inside the plasma membrane or the cytoplasm. The mechanism of the Gram-Stein reaction, we have already seen that in the previous chapter, that how it works. The shrinkage of the pores of the peptidoglycan layer of the gram-positive cell wall actually restrict the crystal violet to escape from them. So what happens that when you actually treat the cells with the crystal violet and the mordant iodine, they form a complex, and it precipitates. This complex is essentially stays inside the gram-positive cell wall due to these pores, which actually constrict and doesn't allow these dyes to escape. What happens on the other hand, that in the gram-negative bacteria, you have a thinner peptidoglycan layer, and large pores of the gram-negative bacteria do not prevent the loss of crystal violet when washed with the alcohol. And alcohol also may remove and extract some of the lipid from the outer layer of the gram-negative cell wall, making the crystal violet dye removal much easier as well. And then you can counterstain it with the safranin. So the cell wall and osmotic protection is an important function. So if a cell is actually a bacterial cell is put in a hypotonic environment, the solute concentration outside the cell is less inside the cell. That means there is a concentration of solute is less. That means the concentration of water is more outside. Water moves into the cell and the cell will swell. But what happens, the cell wall actually protects these bacteria from lysis. Otherwise, they will undergo the osmotic lysis, just like an animal cell or a red blood cell. So this is one of the advantages of having a cell wall. On the other hand, what happens that if a bacteria put in a hypertonic environment, solute concentration outside the cell is actually greater than the inside. That means that outside there is less water, inside there is water. Water will leave the cell, and the cell will undergo the process which is called the plasmolysis. That means the cytoplasm will actually shrink. It will not necessarily kill the cell, but it will shrink, dehydrate, and the cell actually may go into a kind of a dormant state. And this is the process of pickling process, where we use the brine or the vinegar to make an hypertonic environment so that the bacteria cannot grow in that environment. And the plasmolysis actually occurs. If there are some bacteria, due to the plasmolysis, their metabolic process actually slows down. On the other hand, what happened? Lysozyme is an enzyme which is present in our mucous membrane, and it is in our tears. In all mucosal surfaces have this lysozyme, and they breaks the bond between the NAG and the NAM. And also the drug penicillin binds the peptidoglycan synthesis that we already talked about. And if the cells are treated with either lysozyme or penicillin, there's an interesting thing happens. So they actually form what you call as a protoplast or a spheroplast. This is also called a spheroplast. because it's round, spherical. That's why sometimes called a spheroplast. This doesn't have a cell wall. Now, if we just actually put this in a hypotonic media, that means that water concentration outside is more, solute concentration is less. Water is going to move from outside to inside, and the cell will swell. And eventually, the cell will have so much water that the plasma membrane will not be able to hold it, and it will lyse the cell, and the cell will die. This is a typical condition that actually happens in animal cell. Say you take a red blood cell and put in distilled water. This is what is going to happen, which we call as an osmotic lysis. The cell wall prevents this process, but we can use this lysozyme or the penicillin. Lysozyme is a natural material that is present to prevent bacterial infection. Penicillin is a drug. They all do the same way to kill the bacteria. So the cell that lose a cell wall may actually survive in isotonic environment, protoplast, spheroplast. On the other hand, the mycoplasma is a very unique type of bacteria, which is pleomorphic, can have different shapes. And the reason that they are pleomorphic, they never produce a cell wall. That's why they are kind of pliable. And the plasma membrane is actually more resistant to osmotic pressure in mycoplasma, but they are not rigid, pliable, and pleomorphic. Very, very small, about 0.3 micron in size. The next part that we are going to talk about, the cell envelope, the component outside the cell wall, and the outermost layer of the cell wall, capsule, slime layer, glycocalyx. We are going to talk about this glycocalyx and the S layer, and we are going to explain what are these. So the first one is the capsule. So the capsule are well organized and cannot be removed easily from the cell, usually composed of polysaccharides, which are neutral in charge, not positive or not negative, and visible in light microscope, have protective advantages. So what happens that it resists phagocytosis if there's a pathogenic bacteria which is having a capsule. Our phagocytic cells, which are the neutrophil and the macrophage, they will not be easily able to phagocytose them. They also help them protect from the desiccation in the environment if it is too dry. Sometimes they can actually utilize and use the polysaccharide as a food or energy source if the environment is harsh. On the other hand, the viruses and detergents are also cannot enter into the cell or come in contact with the cell due to this capsule layer. So these are all protective functions. In this picture, what you are seeing that here the slide is actually uniformly stained by a negative stain. And you can see that the dot in the middle of this white is the bacterial cell, whereas the white part that you are seeing, which is unstained, and that's why it is completely distinguishable, and that is your capsule. Why it is white and it is not stained by any of these dyes? Because it is composed of polysaccharide, which is neutral in charge, and no positive or negative dyes actually bind to it. No basic or acidic dyes will bind to it, so it is colorless, and the background shows, only the background shows. Now, similar to capsule, the slime layer are diffused, unorganized, and easily removed. And sometimes these slime actually also help in the motility of the bacteria as well. On the other hand, if we just look at this word right here, is that what is the glycocalyx? Glycocalyx is a generalized term which actually includes the capsule and the slime layer. So capsule and slime layer all together is a glycocalyx, and it helps attaching them onto the solid surface. They form biofilm in plants and animals. Say, for an example, the plaque that is formed in your on your tooth, that's a glycocalyx, and the bacteria that is present in there may have capsules, slime layers, etc. The next one that we are going to talk about is the S layer, and regularly structured self-assembling layers of protein or glycoproteins, and they look like a tile. They are tile-like structure. In gram-negative bacteria, the S layer adheres to the outer membrane. In the gram-positive bacteria, they are associated with a peptidoglycan. And there are several functions for the S layer. It protects from the ion and the pH fluctuations, helps in osmotic stress, maintain shape rigidity, promotes adhesion to the surface. And also, this is actually utilized for the potential use in nanotechnology. So this is your S layer. Some of the bacteria do have these. So the next part that we are going to talk about is the bacterial cytoplasm and what are the structures present in the bacterial cytoplasm. Cytoskeleton, intracytoplasmic membrane, inclusion, ribosome, nucleoid, and the plasmid. And we are going to talk about each one of them. So the protoplast is the plasma membrane and everything within, whereas the cytoplasm is the material bound by the plasma membrane. So here is a print mistake, material bounded by the plasmid membrane. This is a mistake. This will be cytoplasm, material bounded by the plasma membrane. So the bacterial cytoskeleton, actually initially this was not discovered, and the scientists actually thought that the eukaryotes only have cytoskeleton and the bacteria actually don't have any cytoskeleton, but that proven to be wrong. The homologs of all three eukaryotic cytoskeletal elements have been identified in bacteria. And these are the homologs or analogs of actins, microtubules, or intermediate filaments. Actins are made up of, these are the microfilaments, or these are actin. Microtubules are made up of protein tubulin in eukaryotes, and the intermediate filaments are made up of myosin in eukaryotes, and there are similar analogous structures in the bacteria as well. So what they do, they actually participate in cell division, localize proteins, determine cell shape. And here there's a couple of examples. For example, this FTSZ in many bacteria forms a ring during the septum formation of the cell division. So these two bacteria, this was a single cell, are dividing into 1 into 2, 1 into 2, 1 into 2. So dividing, and you can see that right here, this green fluorescence is due to the FTSZ, which is tagged with this fluorescent dye. So this is going to form the septum and divide these cells into 2. The FTSZ is very close or similar or analogous to the tubulin protein, which forms the microtubules in the eukaryotes. On the other hand, you can see that this MREB or MBL, these are the proteins which are actually present in many of the bacillus, maintains the shape by positioning the peptidoglycan synthesis machinery, mainly for the bacillus. And these MREB or the MBL, these are closest to the actin cytoskeletal element of the eukaryote. On the other hand, what you will see, that cress, or sometimes called cressetin, this cress is actually rare, and this maintains the curved shape. And these cress is actually very close to the intermediate filaments. And the intermediate filaments actually form some specific proteins which are called laminin or keratin, very common in the eukaryotes. So next, what we are looking at is the intracytoplasmic membrane. So think about that we talked about that the controversy of the eukaryotes and prokaryotes. Prokaryotes, bacteria are one of them, along with the archaea, if we just think that way, cannot have any organelle. On the other hand, what we are seeing that there are some intracytoplasmic membranous structure, but they are not separate. They are actually enfoldings of the plasma membranes. And these are observed in many photosynthetic bacteria. And these stacks of membrane actually are called thylakoids, Thylakoides, thylakoids. And if you recap, that the plant cells have the chloroplast, and these chloroplasts actually have these stacks of membrane which actually contain the photosynthetic pigment. Those are called the thylakoids as well. And these are the analogous structure compared to the eukaryotic thylakoid. And these are found in the photosynthetic bacteria like cyanobacteria. On the other hand, there are some bacteria with a high respiratory activity also do have these stacks of membrane. The reason being that the respiratory enzymes are actually associated with this membrane, and that aids in the high respiratory activity. So the membranes just simply acts as a place to hold those electron transport or other enzymes in a particular position. So the next what we are going to talk about is the inclusions, which are the granules, crystals, or globules of organic and inorganic material that are stockpiled by the cell for future use. And some are within the single layered membrane, and these membranes can vary in composition. Some made of protein, some contain lipids, and sometimes these are referred as microcompartment as well. But they are not really like as developed as the eukaryote. So what kind of ingredients or the storage inclusions the bacteria do? They actually store nutrients, metabolic end products, energy building blocks. Glycogen is a classic example of that. So the glucose is converted to glycogen, and this is stored as an energy molecule. Carbon storage is usually with this polybeta hydroxybutyrate, or the PHB, and these are actually put in a typical structure. You can see that this PHB, or polybeta hydroxybutyrate, is inside, which is covered by layers of proteins. And this is a storage structure or the inclusion in many of the cells. Now, this particular structure or the storage of the carbon is actually of interest to the scientists, because a lot of scientists are trying to develop the biodegradable plastic by using this polybeta hydroxybutyrate and similar kind of structure as well. So the phosphate can be stored as a polyphosphate in granular form as well. And here you can see that the sulfur globules are present in inclusion inside the cell as well. And the sulfur globules are actually essential for the electron transport chain and many of the energetics of the cell, or many of the reactions involving energetics of the cell. And amino acids can be stored as cyanophysin granules. These are pigment granules, and amino acid can be stored in this way as well. So these are some of the example of the different inclusions that can be present in the cell. So the microcompartments, when we are going into that the microcompartments are not bound by membranes, so they are really not true organelles as such. So what they do have, classic example of one of these microcompartments is a carboxysomes. And these are present in the carbon dioxide fixing bacteria. That means that these are autotrophs which can fix carbon dioxide by either chemical or photosynthetic method. So what you have that towards the outside, they have this carbonic anhydrase enzyme, which actually helps the carbon dioxide from the carbonate as such. Bicarbonate HCO3 minus and carbon dioxide, that reaction, carbonic anhydrase, is present near the outside. On the other hand, in the inside, it contains the enzyme which is called the ribisco, which is the ribulose bisphosphate, carboxylase, and oxygenase. Next, the gas vacuoles actually found in aquatic photosynthetic bacteria and archaea. And these are actually helps the buoyancy in the gas vesicle. Just think about that. This is a swim bladder of the bacteria, just like the fish. So this is the function for the gas vacuoles. So the next, what we are looking at, the magnetosomes, these found in the aquatic bacteria, and they are made up of either magnetite, which is an oxide of iron, or the greigite, which is sulfur conjugated to the iron, greigite or magnetite particles. And they are taken up by the bacteria, and they orient themselves to the Earth's magnetic field. And the cytoskeletal protein MAMK actually help to form these magnetosome chains inside the bacteria. Until and unless they are actually in this chain form like a magnetosome, they cannot align themselves to the Earth's magnetic field. So what is the function for this? Now the function is that due to this magnetosomes, it can help these bacteria to find the proper depth where they can find the nutrients, or find the proper sediment where they can find nutrients. So these are some of the function of the magnetosome. The next structure that is present in the cytoplasm is the ribosomes. This is the universal structure for protein synthetic machinery. And this is a complex of protein and RNA structure. So the bacteria and the archaeal ribosome are 70S in size. So let's take a look at what they are composed of. You have a smaller subunit, which is a 30S subunit made up of ribosomal RNA and protein. And you have a large ribosomal subunit made up of RNA and protein. This is a 50S subunit. During the protein synthesis, they come together and form the 70S ribosome. See that the 30S and the 50S, these are not additive to 80S, but this is 70S, which is a little bit smaller. So the S actually stand for the Svedberg, Svedberg. This Svedberg coefficient is basically a sedimentation coefficient in an ultracentrifugation machine. So what we mean by that when you actually try to isolate these subcellular organelles, we actually crush these cells, make a supernatant, and we try to use centrifugation to create artificial G so that these actually precipitate in a certain portion of the tube that inside which we are centrifuging these samples. So this Svedberg unit, or the sedimentation coefficient, gives you a relative idea that where and when these structures are going to precipitate. So just as overall general, that if you have a larger S value, that means it will precipitate faster than a smaller S value. But when they are combined together, you can see that it is not an additive value. Does not mean that they are completely additive like a molecular weight or something like that. So this is just to keep in mind that what is the S value, which is the Svedberg unit, which actually expresses the sedimentation coefficient in an ultracentrifugation isolate these structures. So the bacterial ribosomal RNAs in the smaller subunit actually have a 16S subunit. So if you just look at this, that the small subunit is made up of 30S. Out of that, the 16S is from ribosomal RNA. Rest of it is from the protein to make it a 30S unit. Whereas the large subunit is 50S, made up of 23S and 5S. These are the two subunits of ribosomal RNA, and the rest of the 50S is made up of protein. And these together make a 70S bacterial ribosome. The nucleoid is usually not membrane bound like eukaryotes, with a few exceptions. And here is where the location of the chromosomes are and the associated proteins. Usually the bacteria will have one closed circular double-stranded DNA molecule, just one chromosome, but sometimes the bacteria can have a linear DNA molecule, like a Vibrio do have a linear DNA molecule. On the other hand, there are some bacteria which has multiple copies of the chromosome or more than one. Say, for an example, we actually was talking about constantly with the very large bacteria, E. fisheloni. E. fisheloni have tens of thousands of copies of the chromosome. And what is thought the reason behind that sometimes these large bacteria can actually, their chromosomes can actually get damaged from the ultraviolet and other environmental conditions. And the multiple copies will allow them to have at least some conserved, undamaged part of the DNA for their survival. These DNAs are also sometimes undergo supercoiling and with the nucleoid proteins. But one of the important difference is that in the eukaryotes, the supercoiling of these DNA molecules actually is done by the histones, which actually forms the nucleosomes and form the chromosomes from these nucleosomes. But in the bacteria, you do not have these histone proteins. The proteins are entirely different. So it's a very, very big difference in between the eukaryote and the prokaryote or the bacteria, where there is no histone in bacteria. Plasmids, on the other hand, are the extrachromosomal DNA. What do we mean by that? That extrachromosomal means that you have a very large chromosome, usually either circular or linear, doesn't matter. But the plasmids are separate from the chromosome. These are small double-stranded molecules, closed circular DNA molecules, and they exist and replicate independently of the chromosome. Sometimes they may integrate into the chromosome and they can actually cut themselves out of the chromosome and can come out. These are sometimes called the episomes, and these are inherited during the cell division. So what are the advantages of these plasmids? So think about that plasmids often actually do contain virulence genes, which actually code for toxins or say antibiotic resistant pumps. So these kind of thing. Now, what happens that these are carried by the bacteria. Sometimes one bacteria may actually meet another bacteria of the close species or the same species, which actually don't have that plasmid containing the virulent genes. So the bacteria 1 can actually connect to the bacteria 2 and transfer these plasmid to the bacteria 2, which was earlier naive, now is converted into the virulent form by the plasmid, which is going from one virulent bacteria to the naive bacteria. So these are some of the functions for that. So your question could be that why the chromosome cannot transfer from one bacteria to the other. These are really super large, very big. The chromosomes cannot be transferred through the pili, which in between the cells, but they can be transferred in just small forms or like a part, like the episomal form from one bacteria to the other. And the classification by the mode of existence, spread, and the function, we have different types of plasmids as well. We're going to talk about that in a different chapter. So on the other hand, many bacteria have external structure for attachment and motility. And we was constantly talking about in the previous that pili, and we're going to see that what are the pili. These pili are the hollow tubes which can actually form in between the cells or the bacteria, which can actually transfer these plasmid. Along with that, they have some locomotory and anchoring function as well. So the external structure actually extend beyond the cell envelope in bacteria and function in protection, attachment to surfaces, horizontal gene transfer, cell movement. These are classic example of pili and fimbriae, as well as the flagella. Flagella is many times bigger than the cell, whereas the pili and fimbriae is much smaller. So let's take a look at this, what they are. So fimbria is a singular, and fimbriae is a plural, pilus singular, pili plural. And these are short, thin hair-like protein appendages like this, which are about 1,000 per cell, and they can mediate attachment, surface motility, and DNA uptake, these kind of things. So these are small fimbriae or the pili. These are the small. Whereas the longer ones or the large one, these are known as a sex pilus or sex pili. Longer, thicker, 1 to 10 per cell, and the genes for formation on plasmids. So we just talked about that if there are two bacteria, one bacteria can transfer their plasmid from one bacteria to the other by these sex pilus. And this process of transferring the plasmid or the part of the chromosome, never the whole chromosome, part of the chromosome or the plasmid, and this transferring of this process or exchanging the genetic material is known as bacterial conjugation. And we're going to talk about that in a different chapter. But the bacterial conjugation is much like a similar kind of sexual reproduction of the eukaryotes. So next we are looking at the flagella. Flagella is much larger and very complex structure, and they have different types of varieties how they are actually attached onto the cell surface. Thread-like locomotor appendage helps in motility, swarming, attached to surfaces, maybe virulence factors sometimes. And the pattern of flagella, there are many. Let's say, for example, if they're attached to one end, they are called as a monotrichous flagella. On the other hand, if you have one end, you can also call them as a polar flagella. Polar means only in one pole. It's not shown here, but if you have a flagella in one end and the other, we call them amphi. Amphi means both. Amphitrichous flagella is one flagellum at each end. On the other hand, a lophotrichous flagella is if you have a bunch or a cluster of flagella at one or both ends of the bacteria, we call this as a lophotrichous. These are very important for your quizzes as well. On the other hand, if the bacteria actually contain flagella surrounding the whole structure, we call this peri. Peri means around. Peritrichous flagella spread over the entire surface of the cell. So now let's take a look at the bacterial flagella's ultrastructure, and it is extremely complex, and still this is presented in a very simplified format. It's a thin, rigid protein structure, cannot be observed with a bright field microscope unless specially stained. Like the previous pictures, these are not electron microscope, but here to visualize the flagella, you have to deposit some heavy metals onto them to make them thick so that you can actually visualize them. Very special staining technique. So you have mainly three parts of the flagella. You have a filament right here, which extends beyond the cell and very long. Then you have a hook structure right here, and then you have a basal body. So the basal body is anchored into the cell envelope, which could be the plasma membrane, the periplasmic space, peptidoglycan layer, and the outer lip polysaccharide layer of the gram-negative bacteria, or it can be anchored into the plasma membrane and the peptidoglycan layer. These are the three different parts: filament, the basal body, and the hook. Now, this basal body in the gram-negative bacteria has multiple rings. So you can see that L ring is present in the lipopolysaccharide layer, the P ring is present in the peptidoglycan layer, and then you have a MS ring. This is actually called the membrane spanning ring, MS ring, and the C ring is a cytoplasmic ring, is extended towards the cytoplasm. So you have a L, P, MS, and the C ring, 4 rings mainly in the gram-negative bacteria. Whereas what you are seeing in the gram-positive bacteria, you just have an anchored structure into the plasma membrane and the peptidoglycan layer, a much simpler arrangement of the flagella in a gram-positive bacteria. Flagellar complex process involved actually many genes and many products. So what happens that the new flagellar molecule are actually produced by the ribosomes, and as the ribosome is synthesizing the molecule, it actually goes out. The flagella do actually contain a hollow core, and this type 3 secretion system actually moves these flagellin proteins from the synthesis all the way up to the top, and there's a self-assembly and extension of this flagellar filament as well. And this is how the flagella is actually formed. So the motility, the flagellar movement, swarming, spirochete motility, we are going to talk about a couple of different these things. So when you are looking at the flagellar movement, it rotates like a propeller, and a very rapid rotation up to 1100 revolutions per second. Think about that. This is generated by just only the movement of the protons from the periplasmic space back into the cell, and proton motive force does that. So in general, the counterclockwise, if it's counterclockwise rotation or CCW, causes the forward run. It's caused a forward run. On the other hand, a clockwise rotation will actually cause them to tumble, disrupt the run, causing by the cell to stop and tumble. So these are some of the ways that the bacteria actually move either towards an attractant or move away from a deterrent. So here we are looking at the mechanism of the flagellar movement, and the flagellum is a two-part motor. It's a two-part motor and producing the torque. So the first thing that what we have right here in the middle part is the rotor, and the side part right here, that's the stator. This is the stator right here in the rotor, just like a machine that we actually electrical motor. So the rotor is made up of this C ring, and this actually do contain the Fly-G protein ring and the membrane spanning ring. This actually turn and interact with the stator. And how this actually works. The stator is made up of these particular protein, MOT A and MOT B. MOT A, MOT B, these are the stator proteins. They actually form a channel through the plasma membrane, and protons move through this MOT A and MOT B channel using the energy of the proton motive force. So what happened that right here, this is the periplasmic space, and there are plenty of electron transport proteins which are present in the plasma membrane. These electron transport chain proteins actually move these protons, or the H+, from the cytoplasm into the periplasmic space. By moving these proteins actually, which are not shown here, they form a gradient of proton. More proton in the periplasmic space, less proton in the cytoplasmic space. Now, what happened that these two MOT A, MOT B, these channels allow the protons from this high concentration to fall into the low concentration into the cell. While the proton is falling into the low concentration of the cell, this is actually pushing the revolving door, and that energy is given to this motor to run and form the movement of the flagella. So this torques the power rotation of the basal body and the filament. So the proton motive force, or PMF, actually drive the motor of the bacterial flagella. Very, very complex nanomotor. And here you can see at the bottom, this is a real picture of a cryo-electron microscopy in detail that how a motor looks like. Fascinating structure. The swarming is a typical type of behavior. This occurs on moist surfaces as a typical group behavior by the bacteria. And the most swarmers have peritrichous flagella, and the production of molecule that aid in movement is a typical. So from a center, you can see that they actually move towards this typical flower-like structure, and this is called the swarming motion. The next one is a very important aspect of spirochete motility. We talked about this spirochete at the very beginning, depending upon the shape, and they actually do contain this periplasmic flagella, which is also known as axial fibril or axial filament. Repeating, they contain the periplasmic flagella, which can be called as an axial filament or axial fibril, which winds around the cell. The flagella remain in the periplasmic space inside the outer sheath, outer membrane, outer sheath, and it remains inside. The corkscrew shape actually exhibits the flexing and spinning movement. So how this is actually anchored? So the basal bodies, you can see that the basal bodies are anchored right here, closer to the membranes. And so here there's an anchor and it runs through here. Then there's an anchor, it runs through here, it anchor right here, and it runs through here. So these are just a modification of these flagella. Rather than extending from one end, it wraps around this bacteria, and the movement of these flagella actually cause the bacteria to undulate in a corkscrew-like motion, and that is actually very, very rapid and even help in survival of the bacteria. And sometimes these are actually utilized to bore through the tissue. A classic example that the causal organism of syphilis, which is a Treponema pallidum, the Treponema pallidum actually is a spirochete, and it bores through the tissue by these mechanisms. And remember that the flagella is under the periplasmic membranous structure, and it is not exposed outside, but they are constantly in a dynamic motion or movement stage due to this structure. So there are a couple of different types of movement, like a twitching and the gliding movement. May involve the type 4 pili or the slime. Gliding is through the smooth movements. Pili at ends, short intermittent jerky movement. These can be shown as well. On the other hand, this is actually more important, is a chemotaxis. So thinking about that if the bacteria actually is an environment where there is no chemical attractant or deterrent, then it actually goes straight for a while, then it tumbles, goes in another direction, goes, tumbles, and you can see that this is a random or haphazard movement. On the other hand, say for an example, here you can see that on the green side, this is a bright green side, has a chemical attractant more here, and the concentration gradient from this side to this side is low to higher. Higher green is more concentration. So in this condition, the bacteria can sense the environment by their chemoreceptor and chemosensing systems. And in presence of these attractant here, they will just go into a straight, then tumble, go straight. But what you can see here, that when a chemical is present in a gradient, if it's an attractant, the random movements actually slow down, and they actually travels in a straight direction for a longer period of time. So the tumbling frequency is reduced, runs towards away from the compound are longer. And this behavior of the bacteria is altered by the temporal concentration of the chemical as well. So the last part that we are going to talk about is the bacterial endospores. These are complex dormant structure formed by some bacteria. These are various locations within the cell and resistant to numerous environmental conditions, including heat, radiation, chemicals. And you can see that it can form a central spore, subterminal, terminal, or a swollen sporangium, many different types. So they are extremely resistant to environmental condition, and even these actually cannot be even destroyed by boiling heat. You need to autoclave the samples or the material to get rid of these endospores. So why they are so resistant? So the calcium is present in a large amount, which is complex with this dipicolinic acid. So this dipicolinic acid, and along with these small acid-soluble DNA-binding proteins, which is known as the SASPs, small acid-binding DNA-soluble proteins, and dipicolinic acid interact with the DNA. So what happens, this dipicolinic acid actually insert them within the bases, and these SASPs actually allow the DNA to change conformation. So the DNA in a vegetative cell means actively replicating cell. So just remember that sometimes the term is actually a little bit confusing. So when we talk about the human being in a vegetative state, that means in a dormant state, in a coma. But a vegetative state of a bacterial cell means that it is actually an actively dividing state. So don't get confused by the word vegetative cells. So in a vegetative cell of a bacteria which is actively dividing, the DNA is in the B form of the DNA. What happens that when this dipicolinic acid and the SASP bind or intercalate themselves into the DNA, it changes from the B form of the DNA to the A form of the DNA. A form of the DNA is actually resistant to ultraviolet radiation. So this is actually a very important feature, that the association of these actually can stabilize or make the DNA extremely resistant to radiation and chemicals. They have a dehydrated core, and this just allows the survival of the organism, which actually don't denature all the proteins, but it is sufficiently dehydrated so that it is actually very resistant to different environmental conditions. And you have a spore coat and exosporium on the other hand to maintain its integrity. So let's take a look at the structure. And so what you have, that inside right here, you have the nucleoid or the DNA wrapped up, which is associated with the dipicolinic acid and the SASPs. Then you have an inner membrane right here. Then after that inner membrane, you actually have the germ cell wall, and this germ cell wall is actually made up of peptidoglycan layer. Then you have a cortex, and this is a layer which is a little bit of loose peptidoglycan layer. Then you have a membrane, and you have an exosporium with a lot of different proteins associated in a very complex manner. All these different proteins complexed in a very complex manner give the endospore unique nature, which is extreme resistant to harsh condition. And it has been already found by the scientists. They found some endospores from the bacteria and able to germinate them, which are mind-bogglingly 100,000-year old. This is in your textbook. It is not a science fiction. You can just check your textbook. 100,000-year-old endospores that could able to germinate. So you can easily imagine how excellent this structure is in terms of maintaining its integrity in a harsh condition. We are going to see that how this actually is formed. The formation of the endospore is known as the sporulation. And the process of endospore formation occurs over several hours, normally commences when the growth ceases because of lack of nutrients. And it's a multi-stage process. So what happens, the cell actually divides into two compartments, and you can see that here there is some axial filament is actually formed in this condition, which actually divided the cell into two unequal compartments. And you can see that here in this process, after this axial filament formation, a septum separates. This is called the forespore, and this is the vegetative cell. Now what happens, this vegetative cell actually engulfs the forespore, and the forespore actually is now inside the vegetative cell. In this condition, what happens, the nucleoid of the vegetative cell actually is destroyed, and you can see there are enough amount of enzymes which actually help the formation of the cortex, and it also secretes the exosporium. And then finally, the completion of the coat synthesis increase its refractility and heat resistance, and the endospore is actually freed in the nature. Now, this will stay in the nature for a long, long time, And when an ideal time comes in, it can germinate just like a seed into a new vegetative cell. So we are going to take a look at how this actually happens. So the activation prepares the endospore for germination and often results from treatment like heating process. The germination actually can happen in environmental nutrients are detected. Sometimes if there is water, just simple water can be absorbed by the endospore and it swells, rupture, and increase its metabolic activity to form and it outgrowth is emergence of the vegetative cell, which will actually grow and form the normal function. So the take-home message, the bacteria can have many different structure, but there are some fundamental items that represent virtually all of them. And we actually talked about them in detail. There is a lot of material in there, and just go one at a time and just cover that. You can see that in the book, there is a much more detailed material in there as such, what we have talked in there. But we have touched pretty much most of the important messages that is required for your test and the quizzes and the exams. So that's pretty much your chapter